I'm Liz Zimmer, and I'm in the botany department, and I do a lot of work with DNA. This title doesn't mean we're talking about forensics, but rather a question in plant speciation. And I involved in this project was an NHRE student, and I thought I would talk about this because we've got them in the audience. And I'm doing this work in collaboration with Carl Taylor and our botany lab tech. So the question we're looking at is a question of sequencing for identification in the early land plant, the first vascular plant lineage, the Lycophytes. And Lycophytes, there are three families that are extant. The Isoetaceae, the Selaginellacea, and the Lycopodiaceae. And you can see from the picture of the Isoetaceae, which is what we're concentrating on, that they have a fairly simple morphology and in fact sort of resemble grass, although that's not true of the micromorphology. So in order to identify Isoetaceae species, people have used habitat, megaspore texture, megaspore size, chromosome number, and DNA sequences. So there is some variety of habitat. There are aquatic, amphibious, and terrestrial Isoetes. And the megaspore texture varies in sort of four typical types. We're going to be looking at two species, one of which has echinate or spiky surfaces, and one of which has reticulate or sort of network surfaces. And with respect to the megaspore size, we see that in Isoetes, the size does vary such that species with very high chromosome numbers have a larger megaspore than the diploids. And you can see that we can go up to dodecaploids, actually, which have 132 chromosomes. But what's been really useful in the recent 10 to 15 years are the use of DNA to identify the different species and look at their relationships. And you can see in this slide of the two species we're looking at, Echinospora and Engelmannii, that there are, for a gene called leafy, which is involved in vegetative to floral transitions in flowering plants, and is found all the way back to the bryophytes and even the green plant, the green algae progenitors of the land plants. And you can see here the dots indicate where there are DNA sequence differences, and we have a variety of sequence differences, what are called transitions, which are G to A and C to T, and we have some what are called indels or missing bases, and this leafy gene, we're actually looking at an intron between the coding regions of the gene, and it's very rapidly evolving, and it's fairly long. It's about 1,000 base pairs. So with respect to the diploids, we're looking at divergent evolution with respect to the habitat, the spores, and since they're both diploids, the base number is 22, or 2n is 22, the base number is 11. So you can't really differentiate them by the chromosome number. But what's true in plants is that there is a lot of reticulate evolution where we have diploids coming into contact with each other. In the case of these two species in sort of New England, is where they come in the mid-Atlantic is where they come into contact. And when you have the two diploids hybridizing, often the interspecific hybrid is sterile. And so the way plants overcome this is by chromosome doubling to give what are called allotetraploids. And I'm gonna show you sequence from Isoetes riparia, which occurs in the mid-Atlantic. And we also have sequences for Isoetes etonii, which is a sterile hybrid. So you can see from the habitat is pretty similar in all of the species. The megaspores are larger in the tetraploid, and you see the high chromosome number. So we isolated leafy sequences from the two parental diploids, and because these are hybrids of two genome types, we had to clone the leafy intron region. And what we see in the hybrid and in the tetraploid, we see sequences corresponding to both the Kinospora and the Engelmannii. So that's what you'd expect from a nuclear gene. So the question our intern was looking at last year was determining who is the chloroplast donor, and in many cases that's the maternal parent. So he sequenced leafy genes he sequenced chloroplast spacers, which are rapidly evolving. And what he found was a number of markers where you can clearly see that the hybrid and the tetraploid are closely related to Engelmannii and not to Kinospora. And he was able to look at several accessions of riparia, and all of them had this association with Engelmannii.